All right, today we're going to talk a little bit about eukaryotic diversity. So these are all the eukaryotic creatures that play various roles in microbiology. Now there's a ton of microbe-sized eukaryotic creatures that we're not even going to get to, but we're going to focus in a little bit on a few of the ones of clinical significance and think about some of the differences and similarities between them. All right. So first of all, why do we care about eukaryotic diversity? Well, parasitic infections are in fact a problem, and not just a problem in third world countries, but a problem in the United States. So the CDC notes that more than 300,000 people living in the United States are infected with trypanosoma, which causes Chagas disease. More than 300 infected babies are born every year. For a parasite, that's quite a lot. There are 1,000 hospitalizations for symptomatic cysticercosis. 14% of the US population has likely been exposed to Toxocara, and so many of them are children. More than 60 million people are chronically infected with Toxoplasma gondii. So this one is massively problematic and causes toxoplasmosis, which if you are a person with a uterus, was associated with tampon wearing for extended periods of time for a while, I believe. And then trichomoniasis from trichomonas can cause all kinds of problems. It's considered to be a sexually transmitted parasite. So there are a lot of that. Up here in New York State, we have things living in the lakes, and we have over 2,000 cases of giardiasis caused by Giardia every year. So parasitic infections absolutely occur. They occur in quite a lot of people, and there's a lot of people who think that there's a severe underestimation of these because we don't exactly know what to look for, and they're hard to deal with. But let's go through some of the basic eukaryotes and why we care about them. So most of the eukaryotes that we are going to talk about kind of split into two generalized groups. We've got zooplankton and phytoplankton, and this is how we think about them when they are in water systems, because many of these guys live in water systems. Some of the ones we talk about also come from soils, and some of them live specifically in hosts, such as insects and humans. But the ones that we just run into in the ecosystem are either the animal ones, so zooplankton are protists, they're tiny swimming animals, or the algal-based or photosynthetic ones, and those would be the phytoplankton, because that's how we talk about things that are photosynthetic. All right, but when we talk about ones that live inside of hosts, one of the things that makes eukaryotic protists and protozoa and other things that cause trouble interesting is that they often have a very complex life cycle. So this is just one example. There are a number of different versions of the parasitic life cycle, but essentially what happens is they typically get picked up from the environment. So these are over here shed in feces, and so somehow they end up in your environment, in feces of an animal, or however they end up there, they get on you. And what happens in the environment is they form cysts. So part of the concept, of course, is that the environment isn't really a great place for these to be. It is variable in temperature, it is often variable in dryness, there's not a lot of nutrients, so these things will form little cysts, which are protective coatings that they can hang out inside until things are better. This is very similar to when bacteria create endospores, which is also the same kind of problem, because when things are in cyst or spore form, they're hard for us to detect. They're also hard to kill because they have very tough coatings. Then they enter the human body. Once they enter the human body, now they can say, hey, this is a much better place to be. Let's release the stuff that are from the cysts. And so in this case, we call this the sporocyst, and it releases the sporozoites, which are similar to spores. And we'll get more to spores a little bit later. And this is all asexual. Since it is asexual, that means it doesn't need other things to reproduce. It can just keep making copies of itself, which is very similar to what bacteria do. When these copies become active little guys swimming through the gut system and acting as parasites and stealing your nutrients, we call them trophozoites. So trophozoite is the most common version of whatever's currently living and swimming where it is. They use an asexual reproductive process called schizogony, which creates a whole bunch of, guess what, tiny little pieces. When those tiny little pieces come out, they actually can briefly use sexual reproduction. Now, why would this be important? Well, the key to sexual reproduction is it provides diversity. So instead of just constantly making copies and clones, which something could easily get rid of, parasites have the ability to be a little bit diverse. They have male and female gametes, which can then combine together to create a new cyst. Then when the cyst replicates again, it uses meiosis to split back apart and therefore restart the cycle. So while it's living in you, it's not thinking too hard about diversity. It's just replicate. Then, as it's planning to move to the next place, it creates some different versions, mixes up the genetics, and makes itself more likely to survive, evolve, adapt to the world it's living in. So this whole complicated process really just makes these guys fairly difficult to deal with. They're hard to spot and kill when they are cysts, and then they're easily able to adapt to new environments. So let's talk about the protective properties of many of our eukaryotes. All of them have some slightly different, essentially cell envelopes, the outside portion that is protected. Now, everything that is a cell has some kind of cell membrane, which is our basic lipid bilayer. And it's a little different in each different type. All right, so now our protists are technically animals, and animals don't have a cell wall. So far, everything we've talked about pretty much has had a cell wall. Bacteria have a peptidoglycan cell wall. Archaea have a cell wall made of S protein. But protists don't have a cell wall at all. They have cell membrane, so here you can see the membrane, and a membrane called a plasmalemma. So that is a membrane associated with protists. And what this plasmalemma can do is it can actually pick up and hold on to a series of proteins. So proteins can be very strong. It isn't quite strong enough to be a cell wall, and because it is mostly within the membrane, it's not external to it, we can't properly call it a cell wall. But you can see how it creates a little bit of a protective coating. And in this case, this is a ciliated type of creature. So for example, an actual protozoa is a ciliated protist. And so the little cilia hairs can stick out. They are held onto by the proteins, and it creates a pellicle, which is just a little bit tougher than, like, say, our cells. So since these guys do have to survive as single cells, they have a little bit of protection. Now, both fungi and algae have actual cell walls as part of their cell envelope. So fungi, here's their lipid bilayer cell membrane. It's pretty standard, as is the one for algae. They both look just like the other cell membranes. But fungi have a couple of specific things. They have chitin, which is a sugar polymer, which is very similar to peptidoglycan, but in this case, the connections and the way the sugars are put together is specific to fungi. They're one of the only things that has chitin as a part of the cell wall structure. Chitin is also found in arthropods, but that would be outside of microbiology. And then it's got beta-glucan, so that's very similar also to the bacteria, peptidoglycan, and other proteins to hold it together. So fungi have a pretty strong cell wall. So down here we can see the cell wall structure of an algae. Now they've got a few different possibilities, but the big thing that they have is cellulose. So they have large pieces of cellulose, which again is a type of sugar polymer. So sugar polymers are the main thing protecting most of these. And then some other types of proteins, in this case sulfated fucans, that are all connected. Now this looks a lot squishier than perhaps the tighter fungi membrane, because it is. A lot of times if you think about algae, their membranes have a lot more squish to them, but they're still very strong. Cellulose pieces are long, tough, and fairly difficult to break apart. In fact, we humans can't break them apart ourselves. When cows break apart cellulose, they have 4 stomachs and many bacteria that help them do it. So these make great food sources for something that can break them apart, but not always as great for us. However, they do make great protective layers. So we have cellulose in algae, chitin in fungi, and pellicles instead of a cell wall when we're talking about protists. All right, so let's nail down a couple of protists that are of interest. So the protist, the animal ones, come in a few varieties, and then some of them can switch gears a little bit. Neisseria fowleri is one of the probably most well-known amoebozoid types of protists because it is considered the brain-eating amoeba. So it lives in water systems, and every once in a while, it sneaks through a mucous membrane, finds its way up into somebody's brain, and causes trouble by essentially wrecking shop. It's interesting because it's very amoeboid. So most of the time it's got this sort of clunky, funky type of shape. It can stretch out these little pseudopodia or fake feet to pull itself around. But it also has a form that has some flagella, so it can swim to place, and it can protect itself in a little cyst-like thing, which is, again, why they're pretty tough to deal with. So you have lots of options as protists. They don't have as much external protection, but they have a lot of ways to get around. Their motility is pretty high. So not a lot of amoeba and protists and other things cause us trouble, but Nigeria is definitely one of them. Worms, so helminths, which are actually pretty large creatures, get counted in microbiology because they act like parasites and they have micro-sized pieces to them. So most of the time they lay eggs, the eggs end up in feces, they get into the body, and they follow a very similar life cycle as I was showing you with the protozoa type things earlier. Except that when they grow, they break into something that is much larger. There's two kinds that we would care about in microbiology, the Nematoda or roundworms, and the Platyhelminthes or flatworms. Here's an example of a roundworm, a hookworm. Hookworm gets typically in through soils, and you can see the little roundworm look here, and also you can see how it literally burrows underneath the skin to live, and then it will lay little eggs and then hatch again and cause all kinds of trouble. And then from the Platyhelminthe side, the flatworms, many people are familiar with tapeworms, and tapeworms are, well, exactly that. They cause the kind of trouble that includes them grabbing hold of the intestines, being flat enough to not block up the space and collect nutrients from you. So we count these, even though they're quite macroscopic, as large organisms in microbiology because they lay microscopic eggs and then cause the same kind of trouble as other parasites. Fungi. So we talked a lot about fungi in the last video, so I'm not going to do as much of it here. But as a reminder, what's interesting about fungi, or at least the ones that tend to be human pathogens, is that they often come in multiple forms. So we've got these dimorphic fungi that can have long hyphae and mycelia, so those stringy filamentous bits you associate with fungi, but also bud into these little conidia, which make often little tiny spores. And again, spores are well protected. They tend to float in the air, and they are hard to get rid of. So when the spores come in, they get into the lungs. At that point, the fungi can start to grow again. They will find their way into the bloodstream so they can travel around, and they can be all sorts of trouble. The one bonus to fungal infections is that typically you catch them externally and you cannot pass them to another person. Although the fungi that grow externally on our surfaces, those can be passed from person to person, and Candida especially will travel between people, as well as things the kind of fungi that cause athlete's foot. Fungi, as well as all the previous things, are also still very hard to get rid of. They have very similar cell structures to ours. We're eukaryotes just like them. Killing any of these creatures takes a fair amount of finding exactly the right drugs, nailing them in exactly the right way, and trying to make sure we don't kill too many human cells in the process. Fungal life cycles are very similar to the protozoa life cycles because they can switch between forms of sexual and asexual reproduction. So they can easily make copies of themselves that are essentially clones, which is great for replication and getting out there and doing things, but also they can get some slightly different genetics together to create sexual things and then increase their diversity. Mushrooms are basidiocarps. The ones that you see are mostly the sexual form of the fungi. They're made out of a variety of pieces, and you can see here that generally what happens is the spores fall from underneath the mushroom. So it's talking about the basidia, they're under the bottom of the mushroom. They form these little guys, which can then split up. So zygotes are 2N, but then they split up immediately into multiple pieces. So that is our asexual side. Those can spread out and make lots and lots of small mycelia that are all over the place. So the filamentous part of the fungi is typically asexual. It just spreads out. Then two filaments find each other. If they're opposite mating types, they can come together and then create a new mushroom, which can then create new zygotes and new spores to do something again. The ascocarps, which are the cup fungi, do some very similar things. And these guys tend to be more implicated in sort of the pathogenic side. There are toxic mushrooms, but there's also good mushrooms. Most of the ascocarps you encounter are probably not for eating. And again, they have single filamentous pieces that, if they come together correctly, can form this lovely cup, create zygotes, which then split up and form spores, and the spores are always the part of the fungi that is completely asexual, reproduce on their own, get into things, and cause trouble. So tiny little pieces making clones of themselves is pretty much everybody's way of getting in. Protozoa type things, protists, fungi, and of course bacteria, but they don't have any sexual side. All right, nearing the end, we've got our algae. Now, the cool thing about algae from my main microbial perspective is actually they're probably just as useful as troublesome. So let me tell you about some of the good things algae do. First of all, this reddish algae here is one of the ones that we get all of our agar from. So agar, the structural gelatinous stuff that we use in our plates in our lab, actually comes from the cell walls of algae. This is chlorella, and chlorella is a useful algae because it's one of the very few that produces EPA, which is a fatty acid in the omega-3 category, which is one that is important to developing humans and human brains and human function that is often difficult to get from places that aren't fish and fatty fish flesh. And then this down here is kelp, which looks like a plant, very macroscopic in view, but is actually a brown algae, so it falls into the same category, and it is a great source of iodine, which if you're not having iodized salt, you might need more of in your diet. But it doesn't mean all algae are good. Dinoflagellates can be troublesome. Many of them just live out in the water, but this particular one has a series of neurotoxins that it produces and can create a red tide. So you can see it's got a slightly reddish color or a very reddish color. If you see this out in the ocean, please don't go out there. It is definitely neurotoxic. Dinoflagellates are interesting because they have this very large casing around them, as well as a bunch of flagella. You can't see the flagella in the microscope image, but I promise that they're there. And then finally, the last thing that we discussed from a microbiology perspective are lichen, which is a symbiotic relationship of fungi and something photosynthetic. So perhaps algae, perhaps cyanobacteria, so you can get two slightly different things together. And these are not typically human pathogens in any manner. They often live on trees or on other plants, so they're epiphytes, live on plants. And actually, they can be good sources of food. If you've ever watched the TV show Alone, where they drop people in like the Arctic for a period of time by themselves, they'll eat lichens. Reindeer and other things like that also eat lichens because since they've got something photosynthetic, they make their own food. Then the fungi help protect it, provide moisture, and eat the food that they make, and everybody's happy. But it's a really neat microbiology item that actually grows to a macroscopic size. A lot of these do. So the eukaryotes are always a little bit tough in microbiology because it's like, well, some of it's micro and some of it's macro, but we got to lump them in somewhere. So this is where we do. Hope you enjoyed that brief overview through the eukaryotic microbes. Well, eukaryotic organisms.